Hello everyone, I am Sneha, third BBA student at AIIMS Nagpur, and today we'll be doing previous year questions from the chapter Biotechnology and its applications. So let's get started with the first question, which is in the process of RNAi, the genes they are silenced using which of the following? So RNAi, let us first know about RNAi. So RNAi, it stands for RNA interference. Okay. And how is it done? It is triggered by double-stranded RNA in a wide variety of organisms, including animal, plants, and fungi. So, how is this process carried out? It involves silencing of a specific mRNA. So, there is a single strand of mRNA. This RNA, it is being synthesized in the plant or animal, whatever we are interested in. And there is a other RNA which we are introducing from outside sources. Okay. So these both will then combine to form a double-stranded RNA. Okay. So the double-stranded RNA that is formed by binding of a complementary RNA. This one, which is produced in the organism, this is sense RNA, and the one which we are introducing from outside source, this is known as antisense RNA. Okay, and when these sense and antisense RNA, they bind together, they form a double-stranded RNA, and this double-stranded RNA is actually defective, so there is no translation occurring now. Okay, so it is usually used for pest control and for improving the longevity of the plants or the fruits. Okay, so what are we doing in this particular process? We are introducing a antisense RNA from outside. So antisense RNA is being introduced from the genetic tools using, and this is used to silence the sense RNA so that no further translation occurs. Okay, so we have in RNAi the genes they are silenced using single-stranded RNA, which is option B. Okay, so we have question number two, which is RNA interference. So this is related to the same concept. We have to explain the process of RNA interference. So in RNA interference, it is normally present in eukaryotes. So RNAi, this method is naturally occurring in all eukaryotes. It is a system within the living cells that helps to control the activity of specific genes. So, in this system, we silence some genes, which we are not usually. So, silencing of genes takes place. Which genes? Unwanted genes. Okay. And how do we silence this gene? We silence with the help of mRNA. So, firstly, we have to silence the mRNA so that further protein is not formed after translation. Okay. So, how do we silence this mRNA? We introduce a complementary uh, antisense strand so that double stranded RNA is formed, due to which translation doesn't occur. So, what is the source of this double RNA? It can be either retrovirus or transposons. What is a transposon? It is a mobile genetic material. Okay. So, we have interference, it usually involves silencing of a specific mRNA due to its complementary RNA. So, this complementary RNA, we are introducing it from the outside sources. Moving on to the next one, we have question number three. Consumption of which of the following foods can prevent the kind of blindness which is associated with vitamin A deficiency? So, there nowadays there are new type of cereals and pulses which are being developed which are enriched with nutrients. Okay, so the one which is enriched with vitamin A is golden rice. So golden rice is the species of rice which is vitamin A enriched. And how is this produced? It is produced with the rDNA technology, in which specific genes are being introduced in the host. Okay, so looking at the other option, we have flavor saver tomato. It is also an example of enriched species of tomato. So this particular species is also genetically enriched. But what is the benefit of this particular tomato? It has long shelf life. Okay. So it doesn't ripe or it doesn't get bad very quickly. Then we have BT brinjal. So BT brinjal, they are resistant to BT toxin. Okay, which is present in some insects. Moving on to the next one, we have tobacco plants that are resistant to a nematode. They have been developed by the introduction of DNA, which is produced. That question number four. So we have tobacco plant, and this particular plant it is resistant to nematode. And how is it produced? We have RNAi technology. What is RNAi technology? It is RNA interference. How does RNA interferes? By production of both sense and antisense RNA. If both sense and antisense RNA are being produced, there will be formation of dsRNA. And after double stranded RNA has been produced, there will be no translation. No translation means what? That no protein is produced, due to which the plant is resistant to nematode. Okay. So RNA interference, we have sense and antisense RNA that are complementary to each other. So if this is considered to be the sense RNA, we have antisense RNA. What do they form? They form double-stranded RNA, and there will be no protein. So which was this protein? This was the protein that was produced by the nematode, which infected a tobacco plant. Okay. So what nematode? What is the name of nematode which infects tobacco? So the name of the organism is Meloidy gyne incognita. Okay. So we have, and this particular organism, this affects the roots of tobacco plant. This affects the roots of tobacco plants. Okay. So what is important in this particular discussion? We have the name of the organism that was nematode that was affecting it, and by which method we use it by production of sense and antisense RNA. And what is the name of this particular technology? It is RNA interference. We have a genetically modified brinjal in India. Question number five. GM brinjal. GM means what? It means genetically modified. So this particular brinjal, how is it produced? It is produced by a process known as RNAi. What is RNAi? It is RNA interference. In RNA interference, there is formation of what? Sense and antisense RNA is produced. This sense and antisense RNA they combine together to form double-stranded RNA. Double-stranded RNA would cause no further translation. You know what is translation? Translation is the production of protein from the RNA molecule. Okay. So if there is no translocation, there will be no protein formed. No protein formed, that means silencing of the gene. So there can be different objectives of doing this RNAi procedure in different plants. For example, we have golden rice. Golden rice is a species of rice which is vitamin A enhanced. Okay, so vitamin A enhanced. Then we have flavor saver tomato. This particularly has long shelf life. Then we have GM brinjal, which is also known as BT brinjal. This is for what? This is for insect resistance. Okay. And this modified brinjal, which is modified with genetic process. So this modified brinjal, what is the other name for this modified brinjal? We have transgenic brinjal. And what is the gene that we are introducing in this particular species? We have firstly, we know this is BT brinjal. So, what is the full form of BT here? It is Bacillus thuringiensis. We have Bacillus thuringiensis. Bacillus thuringiensis, it has a specific gene. what is the name of this gene? It is crystal gene, also known as cry gene. Okay, so we take out this cry gene from the Bacillus thuringiensis, and then we have this gene inserted into the brinjal. So this gene is inserted into the brinjal. And how do we insert this gene? It is using agrobacterium mediated recombination. So, agrobacterium mediated recombination. This is the process that is being used, and with this, the gene is inserted into brinjal. Okay. So, and what is the final outcome? That the brinjal plant it develops insect resistance. So this cry protein, it actually cry gene actually synthesizes a protein which is toxic for the insects. This is synthesized in inactive state, and after going into the gut of the nematode, it becomes active. Okay. So this is question number 5. So the genetically modified brinjals in India, they have been developed for insect resistance. Okay. Moving on. One, some of the characteristics of BT cotton. So what does BT cotton signify? BT cotton is a transgenic plant. BT means what? It means Bacillus thuringiensis. So what is the significance of this particular bacteria here? We just read it in the previous question that Bacillus thuringiensis is a bacteria that contains cry This cry it is introduced, BT gene, it is being introduced into the cotton, and it leads to what? It leads to protein formation of cry protein, which is toxic to the insects, and thus cotton is now insect resistant. Okay, and BT genes they were isolated from Bacillus thuringiensis, and they were incorporated into cotton plant to form a genetically modified crop called BT toxin. So, what insect in specific are they resistant to? It is bollworms. So, this BT toxin, BT cotton, it is resistant to bollworm, but the important point is it is of low yield. It shows low yield. So, we have one benefit of this BT toxin, but on the other hand, it has high yield. Sorry. So, it has high yield. So both the benefits are being inculcated into a single species only. So BT toxin, BT cotton is now widely used. Okay. So the option D, the question which is high yield and resistant to bollworms. Moving on to the next one, we have which of the following is true for golden rice? We have question number 7. We know that golden rice is a genetically modified crop. How is it genetically modified? It contains high levels of vitamin A. So, high levels of vitamin A are present in this particular species. And how is this useful? It is a form of rice with biosynthesis of beta carotene. So, in what forms is vitamin A present here? It is present as beta carotene. Okay, and these beta carotene, it usually gives it golden or yellow color. So, this golden rice, the name itself suggests that this golden or yellow color, it is due to high levels of vitamin E. So it is a genetically modified type of rice and fortified food that is produced to combat vitamin A deficiency in areas where this vitamin is scarce. Okay, so where do we get these beta carotene genes from? The genes responsible for synthesis of vitamin A, they are obtained from daffodil plant. Okay, and these are then taken from daffodil plant and inserted into the rice. Okay, so we have next question. Which of the following is true about Bt toxin? Question number 8. We have Bt toxin. So Bt toxin, it is actually from the organism which is Bacillus thuringiensis. And this Bacillus thuringiensis, it produces, uh, it contains a gene which is known as cry gene. This cry gene, it forms cry protein. So when this particular gene is inserted into any plant, there is a synthesis of Bt protein. But this Bt protein, it is in inactive form. Okay. So when this inactive form, it enters into the gut of the insect, it enters into the gut of insect, we get the active form. Okay. So we have option D, which is the correct one. So Bt toxin, that active protoxin. So inactive form is known as protoxin, and it gets converted into its active form in the insect gut. There are several advantages in expressing Bt toxin in transgenic crops. The high level of toxin, it can be very high, so it can make that particular plant resistant to the insects. Okay. Moving on to the next one, we have question number 9. The bacterium Bacillus thuringiensis is widely used in contemporary biology. How is it used? It is used as an insecticide. So, Bacillus, it is used as an insecticide. It synthesizes Bt toxin, which acts as an insecticide. So bacterium Bacillus thuringiensis is widely used. It is gram positive. First of all, it is gram positive. Where is it found? It is found in the soil. Okay. So it produces a paraspore insecticide, which is crystal protein. Gram positive bacteria, they have thick peptidoglycan layer. Okay. So main object of production or use of herbicide resistant we have genetically modified crops. Question number 10th. We have herbicide resistant genetically modified. So GM is what? Genetically modified crops. So what is the use or objective for which we are producing these crops? It is actually to eliminate weeds from the field without use of manual labor. So first point is that we need to eliminate the weeds, the lowest input from our side. So we do not want any manual labor. So without any manual labor, eliminate weeds from the field without the use of herbicides. So we know that there is this particular plant. This is what herbicide resistant. Then we have these weeds. So when we'll just spray herbicide, this is herbicide. So it will kill these weeds. But this plant, as this is herbicide, there will be no effect on this particular plant. Okay. So we need to use herbicides for this particular process. And there is also reduced herbicide accumulation and food particles for health safety. Okay. So what are some examples of herbicide? We have glyphosate. Okay. So they have given answer as A, but in this particular question, both A and D are correct. Moving on to the next one, we have cryendotoxins, which are obtained from Bacillus thuringiensis. So we have Bacillus thuringiensis. It synthesizes endotoxins, and these endotoxin, they are effective against insects. Which insects? Bollworms. What is bollworm? It is actually larva of any moth that affects the fruiting body of a certain crop. So bollworm is larva of any crop, larva of any moth. And what is it affecting? It attacks the fruiting bodies. Okay. So we have option D, which is bollworms. Moving on to the next question, we have question number 20, which is Norman Borlaug, very famous scientist, and he was associated with Green Revolution. Okay. So Green Revolution, what is Green Revolution? It refers to the very substantial increase in the yield obtained by breeding high yielding variety of crop. So there was a substantial increase in yield. Okay. So we used genetically modified high yielding varieties. Okay. So, and he was also, Dr. Borlaug, he was also given the Nobel Peace Prize in 1970. Moving on to the next one, we have again Bacillus thuringiensis, question number 13. This produces cry protein, and this cry protein is actually bioinsecticidal. It is produced as a protoxin. So this Bacillus thuringiensis, it produces protoxin, which is converted to the active form. Where does this conversion takes place? It takes place in the gut of the insect. Why in the gut? Because of the presence of alkaline pH. Okay. So BT cotton, which is a transgenic variety of cotton, has been introduced in India, and it contains a foreign gene. Where is this foreign gene obtained from? It is obtained from Bacillus thuringiensis, and this particular gene, it protects the cotton from bollworm, which is a major pest of the cotton. Moving on to the next one, one of the most important reasons why wild plants should thrive is that they are good sources. Question number 14. So wild plants, they are actually good sources of genes that are resistant to disease and pests. So the wild plants, they thrive under very difficult conditions and with variety of pests infecting them and not enough water, not enough nutrients. So they actually become resistant to all of this, and they are also a good source of genes that are resistant to these. Okay, what triggers activation of protoxin to active BT toxin of Bacillus thuringiensis in bollworm? So, question number 15 is that we have a bacteria. What bacteria? We have Bacillus thuringiensis, and this actually secretes a protoxin. So, this protoxin is ingested by bollworm. What is bollworm? It is actually the larva of insects that feed on the fruiting body. Okay. So this protoxin is ingested by a bollworm, and in the pH in the gut of bollworm, we have alkaline pH. And this alkaline pH does what? It converts the protoxin into active BT toxin. So this BT here signifies Bacillus thuringiensis. So Bacillus thuringiensis, it is a gram-positive, spore-forming bacterium, and it synthesizes Cry1A and Cry2B proteins, and these are toxic against a wide range of insect orders. Moth larvae, that these are known as bollworms, and nematodes, and also human cancer cells. So cryoprotein is effective against bollworms, nematodes, and then we have human cancer cell. Okay, so these are the three particular uh, that it affects. And these toxins, they are produced in an inactive protoxin phase, but they become activated when they dissolve in the high alkaline pH of insect gut. So once they're ingested by the insects, these crystals are solubilized in the midgut. So they are solubilized in the midgut where we have alkaline pH. And these toxins, they are then proteolytically activated by midgut proteases, and they bind to specific receptors which are located in the insect cell membrane, which leads to cell disruption and insect death. So we have Meloidy gyne incognita. It is a pest that infects what? It infects roots of tobacco plants. And how can we prevent this infection? We can be using BT toxin. We can use BT enhanced tobacco plant. Okay. So Meloidy gyne incognita is what? It is a nematode. That is roundworm. So this is a nematode. Moving on next, we have the crops that are engineered for glyphosate are resistant. Glyphosate is actually a herbicide. So these crops are actually herbicide resistant. What is the benefit of these crops? That there will be elimination of weeds without manual labor. And then we have there wouldn't be any accumulation of herbicides in plants. There would be no accumulation herbicide in plants. Okay. So we have options here, which is that plants have been having the broad leaves. They are made resistant to a powerful biodegradable herbicide that is glyphosate, and it disturbs the working of EPSP synthetase enzyme. So if this is taken up by the crop plants, they'll die. We have question number 18. we have in BT cotton, the BT toxin present in the plant tissue as protoxin, it is converted into active toxin. So we have protoxin and it is being converted into active toxin due to the alkaline pH of gut. Okay. So the insect gut, mostly the mid gut, it has alkaline pH. Okay. Moving on next, we have Bt toxins. They are solubilized in alkaline pH of the insect gut, causing death. The Bt toxin protein, it exists as an inactive protoxin, but once it enters into the gut of the insect, it due to the presence of alkaline pH, it gets converted into the active toxin, which causes the insect to die. So this is the mechanism of action of Bt toxin. Okay. Moving on next, we have question number 19, which is golden rice is a genetically modified crop plant where the incorporation is meant for the biosynthesis of. So we have golden rice. Golden rice is actually golden or yellow in color. Why is this golden or yellow in color? It is due to presence of a pigment which is known as beta carotene. So this pigment beta carotene, it is a form of vitamin A. Okay, so this vitamin A is actually necessary for our nutrient requirement. So, golden rice is actually a vitamin A enriched variety. Okay, vitamin A enriched transgenic variety. Okay, so what is the chemical name? What is the biological name for golden rice? It is Oryza sativa, which is a modified crop. It synthesizes beta carotene. So we have next question, which is that pollen tablets are available in the market. Pollen tablets are available in the market for which of the following purpose? So pollen tablets. We have question number 20. Pollen tablets are actually being consumed by some people. And why do people pollen tablets? Because they can act as a supplemental food. Pollen tablets are of high nutritive value, so they act as supplemental food. Okay. So pollen greens, they are rich in nutrients, are used as food supplements. Athletes and race horses, they use these tablets to enhance their performance. Pollen, on average, it contains 22.7% of protein. Okay. So, which of these following BT crops is being grown by by farmers in India? We have question number 21. So, BT crop means what? It has a gene. Which gene? Cry gene. Cry gene is present in these variety. Where do we acquire this cry gene from? It is from Bacillus thuringiensis. So, Bacillus thuringiensis, it produces this cry gene and it is used for resistance to insects. Okay, so BT cotton is being grown by farmers, which is pest resistant. Moving on next, we have question number 22, which is match the following. We have BT cotton, we have adenosine deaminase deficiency, we have RNAi, and we have PCR. So BT toxin, it is actually BT cotton. In BT cotton, we have BT toxin, which is derived from Bacillus thuringiensis, which is option 4. ADA deficiency can be treated by genetic methods, and this whole process is known as gene therapy, which is option A. Then RNAi is present in eukaryotes as a method of cellular defense, which is option number 2. And PCR is polymerase chain reaction. PCR is what? It is polymerase chain reaction, and it is used for detection of HIV infection. That is option 3. So we have 4, 1, 2, and 3, which is our option D. Okay. Moving on to the next one. Which of the following statement is not correct? So the insulin is synthesized in the form of proinsulin in our body. We have question number 23. So proinsulin is into the active form of insulin by removal of C peptide. Okay. So we have to find out which of the following is not correct. So, proinsulin has an extra C peptide, which is not present in the active form, which is correct. Then we have genetically enriched. And nowadays, it is being produced by which bacteria? E. coli. And in man, insulin, it is produced as proinsulin, which is also correct. So the incorrect one is they have given it correct that the functional insulin it has A and B chains. So functional insulin in functional insulin C peptide is absent. So A chain and B chain. But both of these are not actually linked together by hydrogen bond. They both are linked together by disulfide bridges. So we have disulfide bridges present between both of these. Okay. Moving on to the next one, we have question number 4. Which of the following features of genetic code does allow bacteria to produce human insulin by recombinant DNA technology? So, this is a human cell and this is a bacteria. Both of these can produce insulin only when the particular gene is present. Why is this? Because genetic code it is universal. This property of genetic code, that genetic code it is universal, that means what? That everywhere uh, these particular codons, we have A, we have G, we have C. This is a codon. It is made up of three amino acids. So in either in bacteria or in fungi or in humans, at all different places, if the codon is same amino acid would be synthesized. Okay. So the first hormone that was produced by recombinant DNA technology, we have question number 25. First hormone that was produced was insulin, and it was produced in a pig. Okay. Human insulin, and after that, what was produced? Human growth hormone. Okay. Moving on next, which one of the following vectors is used to replace the defective gene in gene therapy? So we have vectors which are used in gene therapy. Example is adenovirus. So adenovirus is usually used in animals, or we can say mammals. So adenovirus, it is an double-stranded DNA virus which causes respiratory diseases. So certain modifications, if we if certain modifications takes place on the fiber proteins, they are used to target adenovirus to cell types, and they can limit hepatotoxicity multiple organ failure. Okay, and it is also used as a vehicle to administer targeted therapy in the form of recombinant DNA or protein. So we have first clinical gene therapy. Question number 27. Gene therapy. First time it was given for adenosine deaminase deficiency. What is adenosine deaminase? It can also be written as ADM. So ADA deficiency is an autosomal recessive disorder that causes immunodeficiency. So it is an autosomal recessive disorder due to which immunodeficiency. Okay. So first clinical therapy, it was given in what? It was given in 1990 to a 4-year-old girl. So this right here is important. The year you need to memorize, it was 1990. Adenosine deaminase deficiency. Okay, what does it cause? It causes immunodeficiency. Moving on to the next one, addition of sugars in fed batch fermentation. It is used to purify enzymes. Okay, so question number 28. We have continuous addition of sugar is done to purify enzymes. Okay. Next, we have consider the following statements about biomedical technologies. Question number 29. We have A, B, C, and D. So, A is during open heart surgery, blood is being circulated in a heart lung machine, which is correct. Next, blockage in coronary artery is being removed by angiography. Computerized tomography, which is CAT, it shows the different detailed internal structure as seen in the section of body. Next, we have X-ray, which provides clear and detailed image of organs like prostate and lungs. So all of these are correct, but we have to tell about biomedical technologies. Which of the following are biomedical technology? So, three main categories are for diagnostic, imaging, and therapeutic. So, option A, which was that during an open heart surgery, blood is being circulated in the heart lung machine. This one is the correct option. And we had option 3, which was computerized tomography. It showed detailed internal structures. Both of these were correct. And we have option C, in which 1 and 3 were correct. Okay. So we have next question. We have question number 30. Ultrasound of how much frequency is beamed into human body for sonography? We have sonography. So sonography is a technique which is used to visualize the structures inside our body. And frequencies do we use? We use 1 to 15 megahertz frequency, which, and what is the most important, where is this ultrasonography mostly done? It is done to show the unborn baby inside the mother's uterus. Okay, so we have next question, which is question number 31. Maximization of animal cell culture technology today is in the production of. So animal cell cultures, they are done for production of monoclonal antibodies. Okay, so monoclonal antibodies are formed, and how are they produced? They are produced by the fusion of myeloma cells, which produce as antigen, and B cells. Okay, so this was the answer for question number 31. Okay, moving on to the next one, we have which of the following is true for monoclonal antibodies? So, question number 32, we have monoclonal antibodies. What are monoclonal antibodies? The monoclonal antibodies, they are identical molecules that are specific for one type of antigen. Suppose this is the antigen, so they are specific for that antigen only. Okay, and how are they produced? They are obtained by injecting the target antigen into a rat or a mouse. Sometimes the spleen cells, they produce antibodies, and these antibodies are then isolated and they are fused with myeloma cells to produce monoclonal antibody. So we have option A, that these antibodies are they obtained for from one parent and they are specific for one antigen. So monoclonal antibodies, they are obtained from one parent and they are specific for one antigen. Moving on next, we have genetically engineered bacteria have been successful in the commercial production of. So the hormone which is widely produced through this process now, it is actually insulin. It was also the first of the hormones that was produced during, using these methods. Okay, so we have number 33, in which insulin is currently being produced from bacteria. Okay, so if we just learn about the structure of human insulin, it is formed by two polypeptides, which is A and B, and they are connected by diamide linkages. Okay, so in 1983, L Lilly was the company. In which year? 1983. Company was L Lilly, and it produced A and B sequences. Okay. Next, we have exploration of molecular, genetic, and species-like diversity for products of economical importance. This is the definition for bioprospecting. So we have question number 34. So let us know about bioprospecting. Bioprospecting is the exploration of molecular, genetic, and species level diversity, or the process of discovery and commercialization of new products. And these products are of economical importance. Okay, we have our next question, which is question number 35. Exploitation of bioresources of a nation by multinational companies without authorization from the concerned company country, it is referred to as biopiracy. So biopiracy, it is the use of bioresources by MNCs. What is MNC? It is a multinational company, and this use is actually without any proper authorization. So there is no proper authorization done. Okay. So What is bioethics? Bioethics is actually the study of ethical issues that are emerging from the occurring in biology and medicine. What is bioweapon? Bioweapon are actually the harmful biological agents that are used as a weapon to cause death or disease, usually on a large scale. So we had option A as biowar. Biowar is what in this particular condition there is a disease producing microorganism, toxins or organic biocides, which is used to either destroy, injure or immobilize livestock, vegetation or a human life. Next we have bioweapon. Already told you about bioweapon, biopiracy and bioethics. So with this we complete our chapter biotechnology and applications. We covered all the previous year questions that were being asked recently from this particular question, a particular chapter. Thank you.